Welcome to Advanced Bioscience Laboratories, ABL, the difference maker, a strategic viral vector GMP contract manufacturer and contract research organization, ready to serve as the catalyst to bring your life-changing innovations to market, partnering with you to save lives. ABL consists of an elite team of scientists with extensive experience in viral vector development and manufacturing. With superb technical prowess and passion, we thrive in the art of solving the complex. We don't stand down from tough challenges and embrace the cutting edge. ABL presents Scalable GMP virus production from bench to clinic. Your presenters today are Jennifer Hain, VP of Business Development and Marketing; Patrick Mayhew, MSc PharmD, General Director, ABL Europe; Beth Thibault, Senior Vice President, Operations.
All right. Hello, everyone. It's good to be here talking with you today. I'm happy to be able to give you some updates on ABL and what is going on in our organization. What we'll cover today is a little bit about who we are. I know for some of you, this will just be a reminder, although for some of others, it might be an introduction. Then I'll hand it over to my colleagues to talk through some case studies of some projects that we've worked on at ABL. And then we'll wrap up with what it's like to work with ABL. So to start with, we have 3 facilities that are currently online. And you'll see at the bottom there that each facility is set up just a little bit different. This is very intentional so that we can take the project coming in and your process and deploy the best tools depending on the project's requirements. So if your project requires a fixed bed bioreactor, we might go to Rockville to place that on the iCELLis. If your process requires larger scale, we would look at either Rockville or Strasbourg. And if your process requires later phase, we would look at either of the facilities in France. Wherever we end up doing your GMP manufacturing, we are set up at that location to support both process development, fill finish, and QC analytical testing. So process development, obviously that's where the process comes in. That can be as simple as a tech transfer and then a move into GMP, or it can be more complicated if the process requires further development and optimization. In either case, the equipment that we have at the sites lend themselves to easy and rapid scale up into the GMP manufacturing suites. Once the product is made and the drug substance is available, we would do drug product fill finish at either the Strasbourg or Rockville sites by having the fill finish capabilities there on site. And then finally, the QC and analytical testing to support release of the product. This here just gives you an overview of who we are and what we've done. So we've supported gene therapies, oncolytics, vaccines, and immunotherapies. We have over 30 years of GMP manufacturing experience and over 20 years of fill finish experience. In that time, we've made and produced over 250 batches. And I won't go through all the viruses and all of the molecules that we've seen and worked with over the years, but you can see an example of them at the bottom. One thing that sets ABL apart is the fact that we have immunologists on staff. So these immunologists are on staff in our Rockville site, and they can work with you to design your preclinical and clinical studies to assess the immune response that your therapeutic has. So a little bit earlier on, prior to GMP manufacturing, but again, we're really able to tell you more about your molecule than any other CDMO. And finally, what's new? So we have expanded capacity, which is very exciting, coming online, and then also some technology updates. So to speak first to the expanded capacity, this site will be based, or already is based, in the south of France, and it will be dedicated to phase 3 and commercial manufacturing. Will enable us to produce therapeutics at a larger scale than we have been to date by being able to scale up to 2,000 liters. On the technology side of things, this iCELLis platform is new to the Rockville facility, and it was brought online last year. And it enables us to operate not only at a larger scale, but also enables us to take open processes and close them so that we can deliver to you a more commercially viable process. Okay, I will now hand it over to my colleague, Beth, who's based in the Rockville facility.
Thank you, Jennifer. As a fully integrated service provider specializing in the advancement of gene therapies, ABL can accommodate a wide variety of production platforms in both adherent and suspension models. Some of the work that we've done to date has been in the areas of MVA, vaccinia, adenovirus, AAV, lentivirus, and others, in a multitude of cell lines, including CEFs, EB66, HeLa cells, Vero cells, A549s, MRC5, and others. We offer a comprehensive service portfolio from tech transfer through process and analytical development and a full range of product release and characterization testing. We have a fully integrated QC development and testing services, and our immunologists are on site to help the client understand their therapy. I will now turn it over to my colleague, Patrick.
Thank you, Beth. Hi, everyone. So I will present 3 examples with an oncolytic project based on vaccinia. In the first case study, so the vaccinia process was developed in a non-GMP environment by the client in North America with roller bottles at lab scale. The client had already initiated a tox study with the product, and they wanted to stay with the roller bottle process. Our proposal was to switch to cell stack or single-use bioreactor, but it has been refused by the client. The challenge was to transform a non-GMP process into a full aseptic GMP process with a GMP material and aseptic validation. The process has been done with splitted roller bottles, RC40, representing a total surface of 17 square meters. Manufacturing operations took place in a grade A/B environment. So the PD work, analytical methods development, and production of GMP material have been done in a fast-track approach. Raw material have been changed, including culture medium, MOI, TPI have been adapted. ABL has been able to produce a first GMP batch 12 months after the tech transfer, including aseptic process simulation, media process tests. As of today, we have produced more than 10 batches to supply phase 1 to phase 3 clinical trial material for this client. The second example is an example to switch to a scalable process. The context was the following. The vaccinia virus process was initially developed with an adherent cell line with ongoing phase 1 and 2 clinical trials. The objective for the client was to adapt the existing process with a limited scalability in cell stacks to an animal-free and scalable process in single-use bioreactor in the perspective of a potential commercial strategy. Additional constraints, the client wanted to keep the initial cell line. Consequently, so the cells have been adapted to suspension with a FBS deprivation. The cell adaptation took approximately 6 months in our PD lab. Then the process has been developed at 10-liter scale and scaled up to 100 liters. The project has been stopped at 100 liters because the proof of concept had been delivered to the client. We reached a reasonable level of yield and purity, and the client didn't want to invest more in this new process until a successful phase 3 or a potential IP. The third example is a design of an adherent process with a very tight timelines. The objective for the client, it was a big pharma, was to be able to provide a clinical material in 12 months. The project was driven by timelines. When the request for proposal has been sent to ABL, the expression system was not yet decided, and the client was hesitating between 2 types of cell line. Based on ABL experience in pox virus, we have provided a risk assessment, a risk analysis to the client on the recommendation. The client has been convinced by the rationale provided by ABL and followed our recommendation. We have produced the master cell bank, the master virus seed. We have developed the process in cell stack at a scale of 24 CA40, what you are seeing in the picture, representing 60 square meters. We have produced 2 engrams and developed analytical methods. The first GMP batch has been produced 14 months after the MSA signature. Once again, and as of today, we have produced and released 6 GMP batches. And once again, ABL has been able to meet client expectations. So thanks to its agility and expertise in post-viruses. For the fourth case study, I will pass my time to my colleague Beth.
Thank you, Patrick. The fourth example was a production platform for an oncolytic indication. This particular program involved the use of a tech transferred process from a previously developed phase 1 indication, in which we received incoming virus bank and cell bank. Those inputs were utilized in a feasibility study to help transfer the process into ABL. Upon completion of that feasibility study, it was discovered that both the cell bank and the virus bank were not robust, and that we were not able to move forward with the program at that time. This included remaking of both the cell bank and the virus bank in order to move the program forward. Once they were completed, we initiated the program again, and we were able to successfully scale up to a 10 cell stack lot size and produce several GMP lots for this program. This material eventually was produced and released to include qualified product release assays and QC testing, and this material was then utilized for a phase 2 clinical trial. Case study number 5 is essentially highlighting our new iCELLis platform. In this case study, we have done a number of optimizations for a lentiviral vector. The goal of this case study was really to identify a process platform for the production of lentivirus utilizing the iCELLis system. In this case, we were able to successfully produce a lentiviral vector first in the iCELLis nano system, and then ultimately scale it up into the iCELLis 500 system. As you can see from the graphs on the left and the right, the iCELLis nano and the iCELLis 500 are yielding similar results at 1, 2, and 3 days post-transfection. These results are very similar to what we have seen in the adherent platform that was produced separately. And this tells us that the adaptation to the iCELLis is quite robust. At this point forward, we are looking to further optimize this model. I will now turn it over to my colleague, Jennifer.
All right, thank you both. So when you choose to place a project with ABL, what you get with us is technical expertise, scientific depth, but also flexibility and collaboration. Patrick spoke to it a bit during his case studies with the agility within which we approach the projects. But we take our clients' feedback, we take your opinion and how you want to move forward with your program. We deploy and apply our tools and our expertise so that we move in that direction together to provide you with a commercially viable process that you can take into the clinic. Thank you for your time today. If you have any questions or would like to continue the discussion with any of us, please leave a note and we will make sure to get back to you. Thank you. Are there any questions?